Welcome back, guys. So far this week we have been talking about the cell cycle, DNA, and chromosomes, mixed in with a bunch of other vocabulary words. Today we're going to be focusing on the M phase of the cell cycle, or mitosis, which is cell division. As you labeled on Monday, mitosis consists of 4 phases: prophase, metaphase, anaphase, and telophase. Following telophase comes a process called cytokinesis, and cytokinesis is what ends up making one cell finally become 2 cells. 2 cells will then go back in the G1 phase, and the cycle continues for the 2 new daughter cells that were formed as a result of mitosis. Yesterday we illustrated how DNA becomes chromosomes during mitosis. Recall that DNA is a loose string, kind of like the yarn we used in class, so that it can be read during G1 and G2 and the S phase when we need to use that to make complementary strands of RNA during transcription, or complementary strands of DNA during DNA replication, which occurs during the S phase. So recall that DNA, loose and stringy, to tighten up to become this chromosome, it wraps around proteins called histones and begins to take this circular appearance here. Notice that there are gaps between these two histones, indicating that it's starting to become tightly coiled, but it's not quite as dense as chromosomes happen to be. At this point, the coiled DNA is chromatin. The process of wrapping around the histones continues, and it gets a little bit tighter, and then eventually we have a chromosome. Notice that this section right here, this entire thing, is taken from this itty bitty box down here in this chromosome. So this chromosome here contains a whole bunch of that. The process of mitosis began around page 140, and I'm going to be bouncing back and forth between page 140 and page 141, going from the text to the image that you can see that is, well, not displayed on the screen. Please follow along with me as I read through this. So our main idea: mitosis and cytokinesis produce 2 genetically identical daughter cells. The combined processes of mitosis and cytokinesis produce 2 genetically identical daughter cells. Follow along in figure 5.7 as you read about the process in more detail below. Interphase. We talked about interphase consisting of G1, S, and G2, so we're kind of familiar with interphase at this point. Interphase plays an important role in preparing a cell to divide. It provides critical time for the duplication of organelles and for DNA replication. By the end of interphase, an individual cell has 2 full sets of DNA, or chromosomes, and is large enough to divide. Keep in mind that if one cell goes in and 2 cells come out that are identical to the original cell, they need an exact set of the same DNA as the original cell has, which is why we undergo DNA replication, but they also need equal numbers of the organelles. So in G1, organelles are actually copied as well. So if the original cell has, say, 30 mitochondria, each daughter cell will end up getting 30 mitochondria. Mitosis. Mitosis divides a cell's nucleus into 2 genetically identical nuclei, each with its own single, full set of DNA. This process occurs in all of your body's cells, except those that form eggs or sperm, and prepares them for cytokinesis. Although mitosis and cytokinesis are continuous processes, scientists have divided them into 4 phases to make them easier to understand and discuss. The 4 main phases of mitosis, in order, are prophase, metaphase, anaphase, and telophase. Cytokinesis, which is not part of mitosis, does happen during anaphase or telophase. Kind of begins during anaphase, finishes upon the completion of telophase. So it's almost something that kind of happens alongside mitosis, but it's not given its own section of the cell cycle. The first stage, prophase. During prophase, chromatin condenses into tightly coiled chromosomes. Each consists of two identical sister chromatids. The nuclear envelope breaks down, the nucleolus disappears, and the centrosomes and centrioles begin to migrate to opposite sides of the cell. Organized microtubules called spindle fibers grow from the centrioles and radiate towards the center of the cell. Pay very close attention to the spindle fibers as we proceed to talk through this. On this image here, this is page 141, you can see the stages of the cell cycle. We have interphase here. Mitosis, we're going to be spending most of our time looking at mitosis. In mitosis, we can see the nucleus right here, and we can see gaps forming in the nucleus wall or nuclear envelope. This indicates that the nuclear envelope or membrane that encloses the nucleus, it's starting to break apart. This allows the chromosomes to actually leave the nucleus where they typically are held. Chromosomes are found in there during all of interphase, but we need to move them out of the nucleus during mitosis. We have centrioles right here and right here. Each set of centrioles consists of two, almost making this T shape, and they begin to move to opposite sides of the cell. I like to say the north and the south poles. And if you look in Metaphase, you can see they're actually in the polar regions, the north and south pole. These little strands here indicate the spindle fibers. Those will be used, as you can see, in metaphase, the second stage of mitosis. So our chromosomes condense. They are able to leave the nucleus because the nuclear envelope begins to dissolve. The centrioles, they move to opposite sides of the cell, the north and south poles, and our spindle fibers begin to form. That is prophase. The second stage, metaphase. In metaphase, the spindle fibers attach to a protein structure on the centromere of each chromosome and align the chromosomes along with the cell equator around the middle of the cell. Once again, we're using those spindle fibers. Those are attaching to the centromeres of each duplicated chromosome. So let's check that out on page 141. Metaphase right here, we can see that the spindle fibers are indeed attaching to the chromosomes. This is a drawn image, so the chromosomes are quite neatly arranged down the middle of the cell. It's not so pretty in reality, but illustrations are illustrations, I suppose. Recall that the centromere is the center of each chromosome where the two chromatids in each duplicated chromosome connect. The spindle fibers attach to the centromeres, and as you look at anaphase, we can see that each duplicated chromosome, consisting of two chromatids, separates the chromatids. So one chromatid from the green one will go to this side, one chromatid from the green one will go to the other side. So metaphase, once again recapping, centrioles are now on opposite sides, chromosomes line up along the middle of the cell, and the spindle fibers attached to the chromosomes. Our third stage, anaphase. During anaphase, sister chromatids separate from each other. The spindle fibers begin to shorten, which pulls the sister chromatids away from each other towards opposite sides of the cell. So let's check out anaphase. The spindle fibers tug kind of like fishing line when a fish is latched, and that tugging action pulls the sister chromatids apart. So one chromatid from the green chromosome goes to this side, the other green one goes to that side. Keep in mind our end result is two new cells. Each of them has to have one of the original chromosomes, which is why we make a copy of these back in the S phase of the cell cycle. So anaphase, short, sweet, chromatids get pulled apart and they begin to move to opposite sides of the cell. The fourth phase, telophase. In telophase, a complete set of identical chromosomes is positioned at each pole of the cell. The nuclear membranes start to form, the chromosomes begin to uncoil, and the spindle fibers fall apart. We can see here that the chromosomes begin to uncoil Remember that loose, stringy yarn? Well, by the time the cell gets back to the G1, which it's almost there, telophase is followed by G1, we need to start using that DNA to make RNA, and then eventually more DNA, and then more RNA again when it gets back to G2. So let's check out telophase. Telophase is down here. We can see that these chromatids in anaphase are now fully on opposite sides of the cell. This cell is starting to get pinched in, kind of like you're pinching a balloon. It will go to two sides, form two little bubbles. We have our nucleus starting to regenerate on each side, and conveniently, the chromatids from each cell are located inside those chromosomes, as they should be. That's telophase. Cytokinesis. Cytokinesis divides the cytoplasm into two cells and completes a full stage of the cell cycle. Cytokinesis differs in animal and plant cells. In animal cells, the membrane forms a furrow or trench that is pulled inwards by tiny filaments, like a drawstring. Gradually, the membrane pinches closed, forming a separate cell around each nucleus. So we can kind of see cytokinesis starting here. These are the furrowings I was talking about, kind of these indentations, kind of like pinching that balloon. And cytokinesis, continued pinching of the cytoplasm, results in two brand new cells. Each of these has an original copy of the chromosomes, and they're genetically identical.